So hello and good day to everyone. Again, thank you Sir Maverick for explaining to us the overview of cellular respiration and the step-by-step process of glycolysis. So again, as was discussed by Sir Maverick, there are several end products of glycolysis, one of which would be pyruvic acid, or another name would be pyruvate. Well, for pyruvate, which is suspended within the cytosol, it needs to undergo or continue on with the process by participating itself with the Krebs cycle or the citric acid cycle occurring in the mitochondrial matrix within the mitochondrion. So in a sense, it needs to pass through the outer membrane first. Well, of course, to do that, we must understand first its chemical nature. So chemically, pyruvic acid is a large and polar molecule, and given its chemical nature, it cannot penetrate easily the phospholipid bilayer of the outer mitochondrial membrane. Because if you remember the discussion of Sir Patrick in cellular transport, the size of the molecule as well as its polarity would be certain limitations contributing to the selective nature of cell membrane. And in this case would be the membrane of the mitochondrion. But since it is really required in the Krebs cycle, it needs to enter the mitochondria via a specific transport protein known as pyruvate translocase. So that pyruvate translocase can be seen in this particular figure embedded within the outer mitochondrial membrane. So as the name suggests, pyruvate translocase, it is able to translocate pyruvate from the cytosol to the intermembrane space and eventually to the mitochondrial matrix. So it's a type of transport protein specifically and selectively only translocating pyruvate and no other molecule. So of course, as it now transfers pyruvate to the mitochondrial or to the intermembrane space first, of course, which is located between the outer and the inner mitochondrial membrane, it will now participate in the transition reaction or the oxidation of pyruvic acid. So of course, prior to the citric acid cycle, we have here the transition reaction or the oxidation of pyruvic acid. So this figure illustrates to us the link between glycolysis and the citric acid cycle, divided into three major steps. So for the first step, we have decarboxylation. As the name suggests, a carboxyl group is removed and will be given off as carbon dioxide. Now eventually that will lead us to the second step, which is the oxidation of the 2-carbon compound. Now you have to keep in mind that the 2-carbon compound left will be oxidized. Okay, so as you remember the discussion of Ma'am Ross and Sir Maverick, when we say the term or when we speak of the term oxidized, it means that the molecule is losing electrons. Okay, when a molecule is losing electrons, it is being oxidized. Now these electrons in turn will be donated to another molecule, and in this case that would be NAD+. So in this particular figure, as you can see, you have NAD+ here having 2 slots. Those 2 slots will be primarily for those 2 electrons that were removed from the 2-carbon compound. Hence, NAD+ will be reduced to NADH. And in terms of the definition of the term reduced, chemically speaking, it means that the molecule was able to obtain electrons. Hence, you now have NADH. Now, for the last step, that would be the addition of coenzyme A. Hence, coenzyme A joins the 2-carbon group simultaneously. Now, of course, since at the end of glycolysis you have 2-pyruvate, of course, 2-pyruvate will participate in this transition reaction. Hence, you have 2-acetyl-CoA from 1 glucose. Now I know some of you are asking, why is it important to create an acetyl from pyruvic acid? I mean, why do we need pyruvate? Or why do we need acetyl when pyruvate maybe can participate directly to the process of the Krebs cycle? Well, of course, you have to keep in mind that pyruvic acid in itself is not a participant of the Krebs cycle. A participant of the Krebs cycle is acetyl. Hence, it needs to undergo a grooming process. And this is the grooming process that we are talking about. So in this particular slide, we have here the detailed steps happening in the oxidation of pyruvate that of course will lead us to the citric acid cycle. So in here we have some general information. Say for instance, another term for the citric acid cycle would be the Krebs cycle. Of course, named after Hans Krebs, who worked out much of this pathway in the 1930s. Okay, you also have here the complete oxidation of the organic molecule. In this case, that would be glucose. And of course, it generates many NADH and FADH2 molecules. A reason why in this overview of the citric acid cycle, we have here the generation of NADH and FADH2. Well, besides those molecules, which are technically electron carriers, other products of the citric acid cycle would include the release of carbon dioxide molecules and the generation of ATP. Now, since we already now have an overview of the citric acid cycle, or another term again would be tricarboxylic acid cycle, let us now begin to understand the process itself. So again, we now have acetyl coenzyme A, the product of the transition reaction. So before it enters the cycle, it needs to release coenzyme A, leaving us now with two carbons, a two-carbon compound. Now this two-carbon compound, upon entering, it will fuse with another product of the citric acid cycle, that would be oxaloacetate. And based on the number of carbon molecules, it is a 4-carbon compound. So a 2-carbon compound plus a 4-carbon compound will result to a 6-carbon compound, and that would be in the name of citrate. So that is for step 1. Now as citrate proceeds to step 2, okay, citrate would be oxidized. So meaning to say it will lose electrons. And so upon losing electrons, those electrons will be donated to another molecule, and that would be NAD+. Hence, upon the oxidation of citrate, or as citrate is being oxidized, NAD+ on the other hand will be reduced to NADH. Now, in addition to the oxidation and reduction process, a carbon molecule will separate from citrate and will leave as carbon dioxide, hence resulting to a 5-carbon compound, named as alpha-ketoglutarate. So same with step 2, alpha-ketoglutarate will be oxidized. Again, it will lose electrons, and those electrons will be donated to NAD+. Hence, NAD+ will be reduced to NADH. Similarly, another carbon molecule will separate itself from alpha-ketoglutarate and will leave as carbon dioxide, hence leaving a 4-carbon compound. In addition to the similar steps with step 2, this 4-carbon compound will also help in the production of ATP by allowing adenosine diphosphate within the mitochondrial matrix and a freely moving inorganic phosphate to fuse together, hence producing now ATP. And the resulting molecule for step 3 would be succinate, a 4-carbon compound, which will be further oxidized in step 4. So meaning to say it will lose electrons and will be donated to FAD. Hence, FAD will be reduced to FADH2, resulting to malate. Now, malate, similar to succinate, will be further oxidized. Okay, meaning it will lose electrons, and those electrons will be donated to NAD+. Hence, NAD+ will be reduced to NADH, resulting finally to oxaloacetate. And then the process again continues for the other acetyl-CoA. Now, throughout this cycle, it is important for you to take note that the TCA, or the tricarboxylic acid cycle, is a successive oxidative decarboxylation reaction. Again, the citric acid cycle, it's a successive oxidative decarboxylation reaction. Why is it a successive oxidative reaction? As the name suggests, successive oxidative. It's a series of oxidation processes, right? Because as you can see in step 2, in step 3, in step 4, and in step 5, almost all of the steps in the citric acid cycle included oxidation of the carbon compound. Now, how about for the term decarboxylation? Well, specifically for steps 2 and 3, a carbon compound, or rather a carbon molecule, left the carbon compound, which primarily defines the term decarboxylation. Hence, allowing now the complete oxidation of the energy source, wherein in this case that would be glucose. So at the end of the citric acid cycle, you have 2 carbon dioxide atoms released, or molecules released, 1 ATP, 3 NADH, and 1 FADH2 per turn. So technically, these are the products per acetyl coenzyme A. And in total, together with glycolysis, pyruvate oxidation, and the Krebs cycle, the complete oxidation of glucose will result to 6 carbon dioxide molecules, 4 ATP, 10 NADH, and 2 FADH2. Okay? Complete oxidation of the energy source, in this case, glucose. Now, since glucose has now been completely oxidized, some of you may wonder, or may even think that the process of cellular respiration is now done, right? It's now complete. When in fact, the main goal of cellular respiration is not to completely oxidize glucose, but to harvest energy from it. And to completely harvest energy in this process of aerobic respiration, these electron carriers, such as NADH and FADH2, must proceed to the last step, and that would be the electron transport chain. So for you to be able to visualize how the interplay between the electron carriers of the Krebs and glycolysis interacts with, or how it interplays with the electron transport chain, let us take a look at this figure. So of course you have here a magnified version of the outer membrane, the inner membrane, the intermembrane space, and the mitochondrial matrix. So again, as we have discussed, NADH and FADH2 from the Krebs cycle occurring within the mitochondrial matrix, and those of the NADH from glycolysis will participate in these series of membrane proteins located in the inner mitochondrial membrane, okay, collectively known as the electron transport chain. So again, as the name suggests, it's a chain, it's a series of membrane-bound carrier molecules that pass electrons from one another to a final electron acceptor. Now specifically, these protein complexes or these membrane proteins occur in the inner mitochondrial membrane, wherein 4 of them are protein complexes, cytochrome 1, 2, 3, and 4, and 2 mobile carriers, ubiquinone and cytochrome C. Now, similar to ATC of photosynthesis, okay, there is the production of a proton gradient. However, for the case of the electron transport chain, the transfer of electrons is coupled with the pumping of hydrogen ions from the matrix. Now, if you compare the creation of a proton gradient between photosynthesis and in the process of the electron transport chain, in photosynthesis, hydrogen ions are being moved with the help of plastoquinone, another mobile carrier, whereas for the electron transport chain, they are simply being pumped. Okay, but similarly, both create gradients. Okay, they move hydrogen ions against the concentration gradient. Now, for the electron transport chain, we are in need of answering 2 basic questions. So first, are hydrogen ions pumped against or down the concentration gradient? Okay. And second, is it dissipating a gradient or is it building a gradient? To answer that, we must understand the process itself via this figure. Okay. So in this figure, you have the upper darker region, the intermembrane space, and the lower lighter region, the mitochondrial matrix, separated by the inner mitochondrial membrane. Embedded within it are the 4 protein complexes and the mobile carriers, simply membrane proteins. So to begin with the electron transport chain, let us begin understanding the left side portion of the figure. So in the left side portion of the figure, you have here NADH. Okay, both or all NADH coming both from glycolysis and the Krebs cycle. Now in this case, NADH will donate 2 electrons to cytochrome 1, which in turn will pump 2 hydrogen ions from the mitochondrial matrix to the intermembrane space, as you can see in the figure. Now these 2 electrons from cytochrome 1 will be transferred to ubiquinone, which is a mobile electron carrier situated between cytochrome 1 and cytochrome 3. Now this ubiquinone, this mobile carrier, will carry the 2 electrons from cytochrome 1 to cytochrome 3. And in cytochrome 3, as it receives the 2 electrons, it will in turn pump 2 hydrogen ions from the mitochondrial matrix to the intermembrane space. Now simultaneously then, 2 electrons from cytochrome 3 will be transferred to cytochrome 4 via cytochrome C, another electron or mobile electron carrier. So as cytochrome C carries 2 electrons from cytochrome 3 to cytochrome 4, cytochrome 4, upon receiving it, will pump 2 hydrogen ions, again from the mitochondrial matrix to the intermembrane space, and simultaneously these 2 electrons that it holds will be donated to 1/2 oxygen, thus creating water or H2O. Okay, now that is for the pathway of electrons coming from NADH. How about for FADH2? Now, while 2 electrons from NADH were transferred from cytochrome 1 to cytochrome 3, 2 electrons from FADH2, on the other hand, will be donated not to cytochrome 1 but to cytochrome 2. So if you take a look at the figure, close to cytochrome 1, in the face of the inner mitochondrial membrane facing the mitochondrial matrix, you have their cytochrome 2. So it will receive the 2 electrons from FADH2 and will transfer it to ubiquinone, a mobile electron carrier, and similarly, it will undergo the same process with NADH, but it simply bypasses cytochrome 1 and then will transfer it to cytochrome 3. And it is because of this that FADH2 contributes less to the proton gradient. Why? Because the electrons of NADH passes through 3 protein complexes, hence pumping more hydrogen ions as compared to the electrons donated by FADH2, which simply passes 2 protein complexes. Okay? And so that is electron transport chain. It already served its purpose by allowing the last electron acceptor, wherein in this case, oxygen, since we are talking about aerobic respiration, to accept the electrons. However, in addition to that, another goal or purpose of the electron transport chain would be to create a proton gradient, not in the mitochondrial matrix, but in the intermembrane space. A reason why, as you compare the 2 regions, more hydrogen ions are located in the intermembrane space. Now, by the laws of physics, we all know that at some point, these hydrogen ions would need to go down their concentration gradient, or that they need to diffuse from higher to lower concentrations. And of course, to be able to do this, they need to pass through the inner mitochondrial membrane, which is of course somehow impossible due to their charge. But with the help of another membrane protein that allows them to pass through, okay, the proton gradient may diffuse or may go down. And that particular membrane protein is known as the ATP synthase. So at the right side of this particular figure, next to cytochrome 4, you have this protein ATP synthase, a membrane-bound enzyme that allows hydrogen ions to pass through. Now, as the name suggests, ATP synthase, okay, it synthesizes ATP by using the energy of the proton gradient. So as hydrogen ions pass through this protein, this membrane-bound enzyme, it allows adenosine diphosphate and inorganic phosphate, both suspended in the mitochondrial matrix, to fuse, hence producing ATP. Again, as hydrogen ions pass through and go down their concentration gradient. And so as a whole, both electron transport chain and chemiosmosis make up oxidative phosphorylation. And again, for aerobic respiration, most ATP are produced via oxidative phosphorylation. Now, since there is somehow a similarity between the creation of proton gradients between the mitochondrion and the chloroplast, let us take a look at this figure. So structurally speaking, there is a similarity, right? Wherein there will always be a region wherein there is higher hydrogen ion concentration and a region with lower hydrogen ion concentration. And for the chloroplast, the higher hydrogen concentration would be the area of the thylakoid space, whereas the lower hydrogen concentration area would be the stroma of the chloroplast, and in between them, what separates would be the thylakoid membrane. Okay. However, please keep in mind that again, the creation of the proton gradient, the development of the proton gradient, is different for photosynthesis to that of the process of cellular respiration occurring within the mitochondrion. So in summary, we have the following produced ATP. So you have those 2 ATP directly produced from glycolysis. We also have 2 NADH, wherein through chemiosmosis results to 6 ATP. Now, mathematically speaking, based from this ratio, 1 NADH produces 3 ATP. Hence, 2 NADH, we have 6 ATP. For pyruvate oxidation, you also have 2 NADH, thus 6 ATP. In Krebs cycle, you have 2 ATP directly produced by the process, 6 NADH times 3, therefore you have 18 ATP. And lastly, you have 2 FADH2 producing 4 ATP. Thus, mathematically speaking, from this ratio, we can tell that 1 FADH2 produces 2 ATP. Now, another illustration of this would be this one. Okay. However, what differs from this figure to the previous figure from the previous slide would be the number of ATP produced. So theoretically speaking, energy yields are different per species. Say for instance, for bacteria, you usually have 38 ATP, whereas for eukaryotes, theoretically, the energy yield would be 36. Okay? It usually lies from 36 or 34 to 36 ATP. 32 to 36 ATP produced for eukaryotes. Now, specifically for eukaryotes, there are several reasons why net ATP does not usually achieve 32 or rather 34 to 36 ATP. One of the reasons would be if you take a look at this particular figure. Okay? After glycolysis, of course, you produce NADH molecules, right? However, NADH molecules must also pass through the two membranes of the mitochondrion, and it can only do so with the help of certain shuttle systems. Hence, for NADH to pass through, it can either undergo one electron shuttle or another electron shuttle. So these two electron shuttles are your malate, aspartate shuttle, and the glycerol 3-phosphate shuttle. Now for the malate aspartate shuttle, it will shuttle glycolytic NADH to the matrix as NADH. Okay, so it will still shuttle glycolytic NADH as NADH. However, for glycerol 3-phosphate shuttle, it will shuttle glycolytic NADH to the matrix not as NADH but as FADH2. Okay, so from these shuttles, you can now tell that there really is a difference between, or there may be a variety of energy yields depending on the cell of the eukaryote. Because say for instance, for the malate aspartate shuttle, okay, it can be found on, or it may be exhibited by cells of the liver, kidney, and heart, whereas the glycerol 3-phosphate shuttle are those processes or shuttles being exhibited by the muscle and brain cells. Now that is only one reason. Another reason would be due to leaky inner membrane, leaky with respect to hydrogen ions. Thus, the proton gradient or the driving force for ATP synthesis is dissipating. A reason why usually 34 to 36 ATP are produced or yielded for eukaryotes. Now, even though it lies within 34 to 36 energy yields, why is it that the net yield for ATP is 32 in this case? Well, if you go back to the process of glycolysis, you can observe there that 2 ATP molecules were used. So, say for instance, if the total energy yield of this cell is 34 ATP, you have to reduce or you have to deduct the 2 ATP used in the first 2 steps of glycolysis, hence resulting to a net yield of 32 ATP. But if it is 36, then still deduce 2 ATP used from the 2 steps or first 2 steps of glycolysis, hence resulting to 34. So again, for eukaryotes, the total yield may vary from 34 to 36, but the net yield may vary from 32 to 34. Other information regarding cellular respiration is that it may be interrupted. Say for instance, by 3 categories of cellular poisons that may either block the electron transport chain, it may also inhibit ATP synthase, or may even make the membrane leaky to hydrogen ions. So to better understand this, you may study this particular figure illustrating to you how some poisons affect the electron transport chain and the process of chemiosmosis. So you have their rotenone, you have their cyanide, carbon monoxide, and you also have their DNP as well as oligomycin. Their position within the figure will already tell you what inhibitions or what interruptions they do throughout the whole process of oxidative phosphorylation. Now, all of these are true for aerobic respiration. But how about for anaerobic respiration? Well, by definition, when you speak of the term anaerobic respiration, okay, it uses inorganic molecules as the final electron acceptor. Okay, every or any inorganic molecule except for oxygen. Okay, it may be nitrate, usually used by E. coli or by Pseudomonas, maybe even sulfate or ferric ion or carbonate. Okay, hence anaerobic respiration. Any inorganic molecule as their final electron acceptor except for oxygen. Now, usually, fermentation is under anaerobic respiration since oxygen is absent in the process. However, fermentation is simply an anaerobic condition. Again, it's not anaerobic respiration, it's anaerobic condition. Not really a form of respiration. Why? Because for both aerobic and anaerobic respiration, carbohydrates such as glucose are fully oxidized, meaning to say electrons were fully transferred to another molecule. Thus, what separates fermentation with anaerobic respiration would be First, the absence of the final electron acceptor. And second would be the failure to fully oxidize glucose or the energy source. A reason why for fermentation, it is only a partial oxidation of sugar or of other metabolites, very different to that of respiration. Okay? And second, in the absence of oxygen, okay, for fermentation, what will be problematic would be the supply of NAD plus and FAD plus needed in the process of glycolysis for it to produce ATP. Class, you need to keep in mind that in the process of fermentation, glucose is the main source of their energy or ATP. And glycolysis may only continue to produce ATP if they have continued support or continuous supply of NAD plus and FAD plus. Now, due to the absence of a final electron acceptor, they cannot really regenerate. Glycolysis in itself cannot regenerate its own NAD plus and FAD plus supply. Hence, they need to be oxidized. In this case, the process of fermentation will provide them an alternate source of NAD plus. So in a sense, fermentation is somehow a substitute of both the electron transport chain and chemiosmosis, or of oxidative phosphorylation in general, as it regenerates NAD+. And fermentation may proceed in two forms. So the first form would be lactic acid fermentation, wherein NAD+ is regenerated as pyruvate. The end product of glycolysis is reduced to lactate. So again, pyruvate is reduced, meaning to say it was able to obtain electrons, in this case from NADH. So as pyruvate is reduced to lactate, NADH is oxidized to NAD+, so that there is still a continuous supply of NAD+ for glycolysis to produce ATP. Another form would be alcohol fermentation, wherein NAD+ is regenerated as pyruvate. Again, the end product of glycolysis releases carbon dioxide or a carbon molecule and will be released as carbon dioxide and will be further oxidized to ethanol. So upon the release of one carbon molecule, leaving only a 2-carbon compound, it will be further oxidized, or rather reduced. Okay, pyruvate will be reduced, to ethanol, meaning to say it will obtain electrons, whereas NADH will be oxidized to NAD+, since it will lose electrons and will donate it to pyruvate. Reduced to ethanol, NADH, oxidized to NAD+, so that the process of glycolysis will still have supply for it to produce ATP. So in summary, you may review this particular slide for both aerobic respiration and anaerobic respiration, and respiration in general, its difference to fermentation. Now lastly, of course, we also have to take note of the connections happening between metabolic pathways. So as you all know, cells, any cell within our body, they use many kinds of organic molecules as fuel for cellular respiration. Now, although glucose is considered to be the primary source of sugar for respiration and fermentation, ATP is generated using carbohydrates, fats, and proteins. A reason why for foods such as peanut, we don't only utilize the carbohydrates. We also utilize the fats and the proteins found within peanuts and within any food that we eat. Because for fats, say for instance, its glycerol fatty acids may contribute to the glycerol 3 phosphate in glycolysis, or it may be a participatory molecule in the transition reaction. For proteins such as amino acids, they may participate in glycolysis as pyruvate, or maybe in the transition reaction, it may contribute to the pyruvate molecule being utilized in the oxidation of pyruvate itself, or it may be even a participatory molecule of the citric acid cycle. So in here, in this particular figure, our point simply is that carbohydrates are not the only source of energy, right? Fats and proteins may also be a source of energy as they also contribute to the production of ATP. And so that is all for cellular respiration. Thank you for listening and have a great day.